Well, thank you very much for that nice invitation. It is, or introduction, excuse me. It is such a privilege and an honor to be able to give the Trudy Bush Memorial Lecture. Unfortunately, I didn't take as much advantage of Trudy as I should have at her time at University of Maryland, where we overlapped. I was junior faculty and she was a senior person, and I adored her from afar and didn't take the time to get to know her as well as I should have. So my first thing is an admonition to all the junior faculty in the audience. If there's a senior faculty that you really admire, go get to know them better, and they'll welcome you more than you realize, I'm sure, and so don't let the opportunity pass you by. Obviously, I have the same photo of Trudy that you had there, but I have another one there that's Trudy with Pat Langenberg. Pat Langenberg sort of filled Trudy's shoes at the University of Maryland, and she's still our stalwart for women's health research, so I want to dedicate this lecture to Pat as well. And nothing to declare. So what are sex differences in the brain and what determines them? It's multifactorial, like most interesting biological questions. So hormones, genetics, environment, and experience all determine sex differences in brain and behavior. But in humans, I think we would all agree that environment and experience are probably the dominant variables. They're also impossible to control for. Every one of us has a unique experience and a unique environment, and so we're not going to get a lot of traction in understanding at a detailed level how these might contribute to sex differences in the brain until we understand the biological contributions. So to understand the cultural influences on sex differences in the brain and behavior, we have to understand the biology. And to understand the biology, we have to use animal models. And the beauty of it is that we can use different animal models to ask different questions. Birds are particularly good for questions about seasonality or about language, because they're the only other species that learns their language. Worms and flies are great for their tractability in terms of genetics or tracing specific neurons. Mice, of course, also have tremendous power of genetics. Voles have told us a lot about parenting and pair bonding, because they're one of the few monogamous species, and also in which males show parental behavior. My particular animal model is the laboratory rat. I use the rat because it's been well studied for a very long time. It breeds well in captivity, and also because they have a very strong degree of sexual dimorphism in the brain, which makes them a good animal model to use. So what is the nature of sex differences in the brain? So for discussion purposes, not biological purposes, but for discussion purposes, we can divide them into three general types. The first is what is really truly a sex dimorphism. And the word sex dimorphism is a lot like love. It's overused. The definition has been lost. But what dimorphism means is two forms. Right? So when we have sex dimorphism, it has to be genuinely different forms. And these are things such as in our animal models, their copulatory behavior. Males mount and intramit, females lure does and take on a sexually receptive posture. Parenting in most mammals is sexually dimorphic, meaning that the males do none of it and the females do all of it. Territory is, the males do all the defending the territory while the females stay home and guard the nest and show extreme postpartum regression. Regression in males does not tend to change markedly after offspring are born. The one that most of us are interested, though, in, that we talk about, and has been really the source of the greatest amount of debate, is the sex differences. The sex differences are endpoints that occur along a continuum, with a different average in males and females, but a high degree of overlap. So things in humans that we talk about are this, things like math skills, language skills. Boys are good at math, girls are good at language, but both boys and girls are very good at math and language. In our animal models, we can look at stress and anxiety, we can look at pain thresholds, food preferences. Even in rodents, females prefer sweet things over males, that sort of stuff. And then the third is a more sort of unusual and less appreciated type of sex differences, what we call sex convergence and divergence, which is that sometimes the sexes are trying to be similar instead of different. Because sometimes the biological burden of reproduction has a cost to it that you might want to avoid. One of the good examples of this is parenting behavior in mammals, in which males are not biologically prepped for parenting. They don't gestate, they don't deliver, they don't lactate. So in some species, there have been hormonal modifications to change the brain to get the male to contribute to parenting. In humans, we know that men, after they have children, undergo a variety of hormonal changes, including reduced testosterone and increased prolactin, and that's no doubt for purposes of reducing his aggression and getting him to be more parental. But sometimes, too, there are sex differences that only emerge under duress. We call these the divergent sex differences. So the males and females start out the same under baseline conditions, but they will go in opposite directions during a stressful situation. This has been shown to have opposite effects on learning under stress in animal and human tests, that sometimes females do worse under stress, males do better. Again, it depends on the test and the circumstance. So what are the sex differences in the brain that underlie these different types? Well, just about everything in the brain can be different between males and females. The size of a particular structure or a subnucleus, the number of synapses made between neurons in a particular brain region, the chemical phenotype of neurons, are they excitatory or inhibitory, they're expressed more oxytocin, less vasopressin, or even just the activity and physiology of neurons in a particular brain region at rest. We've taken neurons out of the male and female rat brain and cultured them in a dish and found that just sitting at rest in a dish, they differ in their resting calcium levels, which is related to their excitability. So, to repeat, just about everything can, not always is, but can be different in males and females. Now, how do we measure these sex differences? Particularly if you're interested in anatomical sex differences, just because they're the easiest to quantify, simple, but looking where the light is. We can use MRI in humans, we can use fMRI as well, we can circle the structure and measure how big it is in a living individual, or we can use fixed tissue. This is coronal section through the rat brain, showing you the sexually dimorphic nucleus, which is one of the most highly studied and celebrated sex differences in the rodent brain. And there's the male nucleus is much larger than the female nucleus, and this is a hormonally dependent sex difference. We can get down to individual neurons, and we can measure the length of the dendrites, we can measure how frequently they branch, we can measure how frequently they synapse. So various things that are quite quantifiable. When are sex differences established in the brain? The true sex dimorphisms that I refer to are universally established early in development during the perinatal sensitive period that follows anal differentiation. In primates, we believe this is almost entirely in utero, with very little going on postnatally. In our animal models, it's both pre and postpartum in the pups. Puberty is now being recognized as the second sensitive period. Some people call it the finishing period. There's a lot that happens when the hormones reemerge at puberty that sort of crystallizes what went on early on developmentally. But then there are some sex differences in the adult that are solely due to the adult hormonal milieu, and these are things that would be more in the sex differences paradigm, right? The sort of subtle differences, not sex dimorphism. So genetic sex, we all know this is an easy audience to present this slide to. Genetic sex is going to determine your gonadal sex. XX chromosome complement means an ovary, XY means a testis, due to the presence of the SRY gene. But the gonadal sex is then going to determine the brain phenotypic sex. So the brain is a lot like the undifferentiated gonad, in that it, by potential, it can go towards a, quote, female phenotype or, quote, male phenotype. And analogous to the undifferentiated gonad, the brain has long been considered to be, by default, female. That that's the natural course of events that it's going to follow unless testosterone comes on along and diverts it off its path towards male. I like to think of it as God really intended the brain to be female. The devil came along with testosterone. The beauty of this system, though, is, particularly in our rodents, is that we can then treat that female with testosterone, and we can trigger her brain to become male. So we can really study the process by having this initial trigger, which is handy. So as I said, brain sexual differentiation occurs early during the perinatal period, and we know a lot about this in our rodents. And we know that it's established by the onset of testosterone synthesis by the embryonic testis in the male. So it's going to begin around embryonic day 18. Many people don't think of the hypothalamic pituitary gonadal axis as being mature until puberty. But of course, you all are familiar with the fact that it's fully mature in utero, and the male testis is capable of copious testosterone production. In the rodent, as I said, this starts at the 18, we get another surge of birth, it then goes away and stays undetectable until it comes roaring back at puberty. We call this early period of hormone exposure the organizational period, because this is when we're going to organize the brain into male or female by these differentiation processes. That is then partnered with the activational period in adulthood. So for activational hormones to have their effects in rodents, say to induce male sex behavior, that has to be the appropriate pairing with what went on organizationally. This all occurs during a perinatal sensitive period, which we define by the onset of when the testosterone secretion occurs, and then as the offset is if we give the female testosterone during this sensitive period, we can convert her brain to a male brain. If we go outside the sensitive period, she becomes unresponsive. She is committed to the female path. And because I'm going to be talking about rodents, and I talk a lot about estradiol interchangeably with testosterone, it's just to make you aware, we all think of estradiol as a female hormone, but in a rodent brain, estradiol is the masculinizing hormone developmentally, and that's because it's converted to estradiol in the brain by aromatase. In primates, the masculinizing hormone does appear to be predominantly testosterone, with an unclear role for estradiol in the developing primate brain at this time. All right, so if we want to understand the biological contributions to sex differences in the brain, we're better off starting with the sex differences that are the most robust, that are truly sexually dimorphic. And in rodents, that's copulatory behavior, shown here with the male showing mounting behavior and the female adopting what's called the lordosis posture, which is a sexually receptive, hormonally dependent behavior, which is shown here by real animals instead of cartoons. It actually takes 3 different processes to get a full male versus female brain. One is that we have to have an active masculinization, which is induced by the testosterone being aromatized to estradiol. The other is, for reasons that we still don't understand, we actually have to erase that default female pathway. We have to defeminize. Interesting thought question, why? Why does the male need to get rid of that? We don't really understand. But so those are the 2 processes that actively occur in the male, as opposed to the feminization process, which is a 3rd active process. Because I call it a default process doesn't mean it's not active. It's just, as we said, what's going to happen normally. One of the challenges with feminization is because we don't have a trigger, we don't have something we can give to induce feminization. It's much, much harder to understand because it's a moving target occurring developmentally. And while we might be able to screw it up, it's much more important to be able to try to mimic it to understand what's happening. And we know that there's different brain regions that are controlling these different processes. So for example, masculinization is going to be controlled by a brain region called the preoptic area. It's preoptic because it's pre to the optic chiasm. It has nothing to do with vision. This is the most sexually dimorphic region of the brain. It also contains the GnRH neurons that are going to control the anterior pituitary and gonadotropin secretion. And it has the sexually dimorphic nucleus, and etc. It's a very, very sexually dimorphic brain region. If you lesion it in a male, he loses all interest in sex. And we know that's true in humans as well as in rodents. The medial basal hypothalamus, which is right next door, is the main brain region controlling female sex behavior. And it's under a different set of rules and a different set of mechanisms to mediate that. But I'm going to just drill down more deeply into just looking at the preoptic area so that we can sort of see the power of the biological model to sort of elucidate general principles about what might be going on in the establishment of male versus female sex differences in the brain. So how do steroids change the brain? Well, one of the big ones that I'm particularly interested in is that they change neural circuits. And so they can change the number and type of neurons in a particular region. They can change the size of projections from one region to another. But they also can really change at the very micro level the number and the types of synapses on the specific neurons, the excitatory synapses versus inhibitory synapses. And one of the easiest types of synapses to study is the excitatory synapse on the dendritic spine. Dendritic spines are just what they sound like. They're just little protruberances off of the dendrites that receive synapses from axons, and they are almost uniformly excitatory, which makes them easy to study. So the more dendritic spines you have, the more excitable those neurons are. So we just asked a very simple question. Is there a difference in dendritic spine synapses in the newborn brain of the rodent, looking in that preoptic area? So we just looked at one-day-old pups, we collected their brains, and we quantified their spines. And what we found is that, indeed, there's a big sex difference. Males have over twice as many dendritic spines as females, And what it is that they pack more into a specific area. What you're seeing on the bottom there, that's a western blot for a protein called spinophyllin, which tends to be concentrated in the spine, so it gives us a lovely proxy marker. And what you can see is each one of those bars, that's one male's preoptic area. So we're looking at 5 males on the left and 5 females on the right. It's a visually obvious sex difference. We also use Golgi-Cox impregnation so that we can visualize the neurons and actually count the little hairy protuberances. And as you can see, the male has many, many more packed in that same length of dendrite as the female. This kind of sex difference, this magnitude of differences in dendritic spines, for anybody who doesn't study sex differences, who was, say, looking in the hippocampus before and after learning, they would just do backflips over this. They would not believe it. There's no such thing as that much difference in particular brain regions. In fact, whenever I show this to my colleagues who do real neuroscience, do hardcore neurophysiology, they're just like, that can't possibly be true. You know, they think, do you realize how important that is? Yeah, I realize how important that is. Why don't you guys pay any attention? Well, it's not the hippocampus, it's not the cortex, it's down there in the lowly hypothalamus. But it really shows how markedly different these brain regions can be in males and females. So then, like any good neuroendocrinologist, we have to ask the next question: do hormones mediate that sex difference? So we take the newborn females and we give them estradiol or testosterone, and we show, in fact, yes, it's a completely hormonally driven thing. So within 2 days of hormone treatment, the female shows this massive increase in these dendritic spines and the associated proteins. So the big question is how? What are the hormones doing? This is a really, really potent modulatory effect on brain development. What is the steroid doing? And that's my forte, is to ask these mechanistic questions. So you might see over there on the right-hand side that E plus indo. That indo refers to an indomethacin, which is a drug that many of you might be familiar with because it's a cyclooxygenase inhibitor. And so for reasons that I won't go into, we made the still surprising to me discovery that hormones' effects on the brain are being mediated by an inflammatory molecule, the prostaglandins, in particular PGE2. So prostaglandins are membrane-derived lipid signaling molecules from arachidonic acid. They're cyclized into short-lived precursors, PGG2, PGH2. And then from there, they're sort of finished off with various finishing hormone enzymes into what we know as the prostanoids, one of which is PGE2, made by PGE synthase. PGE2, prior to that, was known for its role in inducing fever. So whenever you have a fever, it's because you've got too much PGE2 in your brain. And when you take an aspirin, you inhibit cyclooxygenase, reduce your PGE2, and your fever goes away. We found that actually that PGE2 is also being regulated by estradiol. So that when we treated newborn females with estradiol for just 2 days, and then measured PGE2 in the preoptic area, they had a 7-fold increase in PGE2. In fact, some of them were undetectable in the control group, and all of them had screaming high levels in the estradiol-treated group. We measured all the prostanoids and didn't see any significant effects or changes in any of the others, so this was highly specific to PGE2. We had to ask the question, how is estradiol increasing PGE2? There's cyclooxygenases, which are the rate-limiting enzymes, but as I said, there are these sort of finishing hormones, and all our money was on PGE synthase as being the site of estrogen action, which is why you have to do experiments, because you have to show sometimes that your hunch is completely wrong. In fact, what we found was that estradiol was increasing both cyclooxygenase 1 and 2, not PGE synthase. So there's a yoking between PGE2 production and cyclooxygenase, and so it appears that there's a shunting towards the PGE2 pathway. We've determined this at both the mRNA and the protein level. This is a very potent regulation of the cyclooxygenase enzymes. So how do we connect brain sex differences to sex differences in behavior? Well, to say, well, why do you care how the estradiol is acting? Is another way of putting that, as long as it's active. Well, by understanding the cellular mechanisms, we can make these causal connections. So we wanted to identify factor X. So is PGE2, in fact, factor X that is going to change the brain and change behavior? The other way we do it is we can block factor X and ask, is that going to block the changes in the brain and block this behavior? So what we did was ask a simple question: do prostaglandins increase dendritic spines in the developing POA? So on the day of birth and 1 day later, we injected right into the brain. We can't give this peripherally because it has to cross the blood-brain barrier. So we injected right into the brain prostaglandin E2. And we waited a day or two, and then we collected the brain and quantified the spines. And to my still surprise to this day, we found that PGE2 exactly mimicked the effects of estradiol. Right? So it induced the dendritic spines to precisely the same degree. It was not additive when put together with estradiol, and it was specific because if we gave PGF2 alpha, another well-known prostaglandin involved in reproduction, it had no effect. We then did the inhibitory experiment and said if we block factor X, if we block prostaglandin production, does that block estradiol's action? And indeed it does. You can see there the vehicles, the control, estradiol plus indomethacin has no effect. We give the PGE2 so that we kind of make an end run around the indomethacin. We show that the cells are still healthy and can respond. We get our full induction of dendritic spines. And indomethacin alone, in this case, has no effect. So we've shown that estradiol increases cyclooxygenase 1 and 2, and cyclooxygenase 1 and 2 increase PGE2. But how do we get from prostaglandin to changes in dendritic spines? So it would take me a very long time to give you all of the experimental evidence behind this, and with apologies to the several graduate students who spent years of their precious lives elucidating this mechanism, I'm going to summarize it now all in one sort of cartoon. Because we really got very, very specific down into the phosphorylation of particular serine residue on the GluR2 subunit of the AMPA receptor. But those details aren't as important as it is in putting together this working model of how hormones mediate synaptogenesis in the developing POA. So we start with testosterone falling out of the sky, it's aromatized to estradiol, where it's going to bind to estradiol's receptors in the cytoplasm, translocate to the nucleus, induce transcription of the cyclooxygenases, both 1 and 2. That's going to increase production of prostaglandin E2. Prostaglandin E2 is going to act on a neighboring cell on the dendrite. It's going to activate the EP2 and 4 receptors, which increases production of cyclic AMP, activating PKA, which we know clusters AMPA receptors at the membrane. Simultaneously, the PGE2 is going to induce glutamate release from astrocytes, which it causes to differentiate. That glutamate is then going to cause those dendritic spines to form and stabilize. So I show you this to illustrate the point of how many steps there are in the process to get to this final differentiated pattern between the males and the females. So there are many, many different points at which things can change. By genetics, by environmental insults, etc. And this is just one mechanism in one brain region. But the real $50 million question is, did we change behavior? I can show you we changed the spines, but did we change the behavior? So if we give factor X, are we going to in fact mimic the effects of the estradiol? Or if we block factor X. So to do this, this is how we study sex behavior in rodents. We put them in glass aquaria, and we pre-treat the females with hormones so that they're sexually receptive. And then we put the males in and we say, do you feel like mating? And you can see here on the top, you're going to see real textbook sex behavior. There's a mount, and the females are dosing, and then she jumps away. On the bottom, what you're seeing, you see that female waggling her head? That's like the sexiest thing a rat can do. So she's like, she is flirting like mad with this male who knows she's there, but is not all that interested. And so we do this for about a 30-minute period, and we can quantify how long it takes the male to mount the female, how many times in a given time he will mount her, and how many times he will intermit, etc. So it's a really nice quantifiable behavior. And you see the group upstairs, party going on, having a great time. Downstairs, he just wants to be friends. And she's about, you see how still she is? She's going to hop and dart and wiggle her head in a minute, and it just absolutely drives male rats wild, and he doesn't care. You can watch her, wait for it, there she goes, it's her best hopping and darting, and he's not interested. So the reason he's not interested is, in the bottom, that was a male who was treated with indomethacin as a neonate, 2 injections, raised to adulthood, no other manipulations. So he's our dud. The stud on top is actually a female. So those are actually 2 females interacting. And that was a female who was injected with PGE2 as a neonate, raised to adulthood, given adult male testosterone. Fully normal male sex behavior, totally indistinguishable from any normal male, other than that she doesn't have a penis. So we showed that this masculinization of the brain occurs within the first few days of life. And so because these don't were not tested until 2 months later. In fact, we even showed that a single injection of that PGE2 has the same effect. So how is this memory of this early life programming maintained? How is it that the brain stays the same throughout life? Well, there we turn to epigenetics. Epigenetics are modifications to the genome that integrate the effects of hormones, environment, and experience, but do not change the DNA sequence. There's two major types of epigenetics. One is chromatin remodeling. This has to do with the acetylation of the histones around the DNA. The other is DNA methylation. It's direct methylation of DNA residues. This is the most fundamental epigenetic process in all of our bodies. It's the reason a big toe is not growing out of your cheek and things like that. So it's how we're differentiated is by DNA methylation. All of these different things have been shown to have sex differences. But in my lab, we've been particularly focusing on the DNA methylation, which can restrict gene transcription. And so what controls DNA methylation, we want to know? It's controlled by enzymes called DNA methyltransferases, or DNMTs. And they are two types. One is for like sort of the static methylation that's going to, again, keep a big toe from growing in the wrong place. And then there's the ones that are sort of in response to environment and experience, and they're called the de novo methylation. However, we're discovering a really interesting thing in the brain. The brain does not follow these rules, and the brain appears to be methylating and unmethylating DNA all the time. So it seems to be a epigenetically privileged environment. What we can do is we can measure the activity of this enzyme in the brain. We use a colorimetric assay. We use a methyl donor. We put in the tissue from males versus females. We look at how the color changes, and we can show that the activity is higher or lower in one sex versus the other. So to remind you again, our question is, how is hormonally mediated organization in the neonatal brain maintained? And is it maintained by epigenetics? How does this early hormonal effect have a long-term effect? And so we just asked, are there sex differences in DNMT enzyme activity in the neonatal POA? And the answer is yes. The females have more DNMT activity, and if we give them estradiol to masculinize them, it's inhibited and comes down to the male level. This is parallel with an increase in overall DNA methylation in the female's POA. So she has more genes that are methylated than the male does. So if we inhibit that methylation, what happens? Will that masculinize her POA? And so what we did was we gave a DNMT inhibitor called DNMT just during those first couple days of life, again raised her to adulthood and said, what's your brain look like? Her brain now looks like a male brain in terms of its neuroanatomy. So we have the normal male given Zebulirin, who looks like the normal male, the normal female, half the amount of spinophyllin. Give her Zebulirin 60 days earlier, and she's got the pattern of a male. Well, is this also true of behavior? You have to do the money experiment and say, is it really changing your behavior? And sure enough, that female who had nothing else done to her but inhibition of her DNMT activity those first couple days of life now looks exactly like a male. She's showing male mounting, male intermitting, she's got male level latencies, etc. She's, again, indistinguishable from normal male. So we have to conclude here that high levels of DNM activity are preserving the default female brain by repressing the male genetic program. So the default is really an active process of keeping that male genetic program at bay. And all the male hormones are doing is actually releasing a suite of genes from repression by the methylation in the female. So what are these male-specific genes? To find this out, we can look at female gene expression and male gene expression, and then female gene expression that is released following DNMT inhibition. And those that overlap are likely to be our male genes. To do this, we used Illumina rat bead arrays, and we did the heat map that I'm sure you've seen before, and we compared females with vehicle, females with Zebulirin, males with vehicle, males with Zebulirin. And so females with vehicle and males with males with vehicle and females with Zebulirin, where our genes of interest lie. And when we did this, we found that there were 23 genes that were increased by DNMT inhibition in females, 17 that were decreased. And these were common. These are our candidate genes that were also shown to be higher in the male or lower in the male versus the female. So we have 35, not that many really, 35 candidate genes to explore. We've actually moved this ball down the field quite a way since I first prepared this talk. So this is where we're going next. So my last couple minutes then, what about the human brain? Is the human brain sexually differentiated? And of course it's complicated, it's not as much as some say, but not as little as others wish. So cognitive endpoints unique to humans cannot be modeled in animals. We cannot model mathematics and language in animals, and so we cannot use these kinds of approaches to ask that question. Sex behavior is also not stereotyped in humans. We don't have mounting and overdosing, maybe some clubs, but we don't. But what about, can we look and get an insight into other cognitive and social behaviors? So one is, this is work Melissa Hines done at Cambridge, just asked, do children have different toy choices? And she has shown, in fact, they do. They have highly dimorphic choices in toys where you give them an array to choose from and they think no one is watching. Interestingly, this is also true in rhesus macaques. This was done in free living macaques at Emory. The one on the left is a little girl who's taken a doll and she's inspecting its genitalia. And the one on the right is a male who has got not only does this car move, but it flashes lights and makes noises when it moves. So can we ask, we use a natural experiment, is this also affected by hormones in humans? And we can use congenital adrenal hyperplasia, which again, thankfully, I don't have to explain to this audience. As you all know, it's the result of elevated, it produces elevated androgens in utero in females that are born with ambiguous genitalia. The question is, are their brains also masculinized? And what Melissa Hines has shown is that indeed, when it comes to toy choice, they are. So what you're looking at is, on the top is playing with girls' toys, and girls with CAH are the blue on the left, and they play with girls' toys less than their unaffected sisters. Boys don't play with girls' toys at all. Then we look at playing with boy toys, and the CAH girls are higher than their unaffected sisters, not quite the level of the boys, but close. And then you can just look at their sex-type activities and interests. Now, it's not an absolute predictor, because if we look at rough and tumble play, this is showing from two different studies put done by Sherry Barenbaum in Illinois, that control girls and CAH girls both show lots of rough and tumble play. There's lots and lots of overlap, but on average, you get slightly more in the CAH girls. Then just moving across the ocean over to Japan, we can also look at spontaneous drawing of the boys and girls to get sort of an insight into their brains. There's a very clever study where they just gave boys and girls paper to draw pictures on, and they then analyzed the drawings. The top object there is moving objects, of which 93% of boys draw a moving object and only 5% of girls do, whereas 97% of girls drew a person. Most of them drew flowers and butterflies. They also looked at their color palette by measuring the crayons when they were done, and showed that the girls used a much warmer palette than the boys. These are some of their drawings. You can see the girls, they have a person, they have a sun in the corner, they have flowers, they have horizons. The boys tend to have very few horizons. They have lots of frenetic activity. As you can see, there's always lots going on. And these are the drawings of two CAH girls. So one has got a car on the top, and the one on the bottom I like because it merges the two. It's got a lot of activity, but also has houses. It's basically a large fish eating a town. It's got lots of color, lots of activity. Very nice. So, and then lastly, my work is funded by the National Institutes of Mental Health, and you might ask, why? Do they really care about the sex lives of rats? No, they don't. They care about the fact that autism is 3 to 4 times more prevalent in boys than girls, dyslexia is 3 times more in boys than girls, ADHD 10 to 1 boys to girls, eating disorders, girls 13 to 1, women to men, anorexia, bulimia 3 to 1, women to men. The whole suite of affective disorders, depression, compulsion, panic, anxiety, 2 to 1, women to men worldwide. Schizophrenia, early onset schizophrenia, 3 to 1 boys to girls, whereas adult late onset is 6 to 1 women to men. And why do I say boys to girls, women to men? Males are at higher risk of developmental onset disorders, all of those, autism, dyslexia, early onset schizophrenia, ADHD, all prepubertal, frequently disorders of wiring, whereas females are at much higher risk of adult onset disorders, which are frequently disorders of neurochemistry, depression, anxiety, panic, etc. So by understanding the basics of brain development, we get insight into these sex differences, and I thank all the individuals who did the work. Thank you very much. I'd be happy to take any questions.
Thank you, Dr. McCarthy, for making so simple a very complex issue. We as endocrinologists take care of patients like also want to go in the opposite sex, transgender, to be treated with sex hormone after approved by the parents when it's minor. Then says later, court, at least in Florida, we follow a protocol, they will very strictly about that. We reviewed the literature in the last 2 years. There is a paper from Holland, I don't recall exactly the author, mothers who smoke during pregnancy perhaps is one of the risk factors that can lead to that, changes in more frequent brain sex.